All right. Thank you very much, Mr. Lamini. Good afternoon, colleagues. As I have been introduced, my name is Mangali Soziane, uh, based at the National TB Reference Lab, also working for Baylor under the sequencing project. I do hope that, colleagues, you can clearly hear me. All right. So today we'll be talking about next generation sequencing. In the context of TB, just like Mr. Lamini has mentioned. So we can go through the objectives of the presentation. Okay, yeah, the objectives of the presentation, colleagues. So we are going to look at key definitions. We are going to look at the NDTB strategy. Then we look at the network. So the sub-Saharan sequencing network. So this is the one which is supporting the implementation of next generation sequencing in collaboration with the stakeholders like Baylor and also the MOH. Then you're going to look at the progress, or like how far, or what are the lessons learned from the implementation of sequencing. Then you are going to review some sequencing results and also just to look at how we can optimize the workflow. So, ladies and gentlemen, you might have seen this slide some time ago. So for us, in terms of TB, it's very important to understand these key definitions. So like drug-resistant TB, which is a term to describe a strain of MTB that is resistant to one or more anti-TB drugs. So some of the key definitions that we normally, that are most common, you can find people talking about monoresistance. You can find people talking about XDR and also MDR. So just to recap from what we did last few weeks. So we say monoresistance, it's when the Mycobacterium tuberculosis is resistant to one of the first-line drugs. So the H is for isoniazid, R for RIF, Z for PZA, ethambutol. So polyresistance, it's more than one first-line drug, excluding combination of isoniazid and rifampicin. So now you're going to multi-drug resistant TB. So this one is when you've got resistance to isoniazid and rifampicin. So why are these two drugs so important? Because these are the main potent drugs for first-line regimen. Then the pre-XDR. So pre-XDR is when you've got an MDR TB plus a fluoroquinolone. So a fluoroquinolone can be moxifloxacin or levofloxacin. Then XDR, it's pre-XDR plus a group of one of the group A drugs. So it can be bedaquiline. Okay, colleagues, let's go to the background. Why are we really here? Why are we really here? So, ladies and gentlemen, in 2014, the United Nations Health Assembly was held. So here they discussed the challenges related to TB. So we can look at the areas where we've made some circles. Let me try to get a pointer here. So, so here in 2014, it was observed that 3.6 million people with TB are missed by our health systems every year. That is the main point I would like to bring to our attention. And also the next point today, we are saying MDR TB remains a public health crisis. So only 1 in 4 MDR cases detected and 1 in 2 cases cured. So when you look at these numbers, it raises some concern. So the World Health Assembly came to a resolution to try to address these challenges, where they've set some goals, like by 2030, this is what is supposed to happen in regards to TB. So they came up with a document like this. So you might have seen this document, the NDTB strategy. So I'll just go straight to what it actually entails. So as I've mentioned, that's the World Health Assembly in 2014. They sat down to discuss issues related to TB. So the aim is to end the global TB epidemic as part of the newly adopted Sustainable Development Goals. So I think this background information is very key to us colleagues to understand that this is not just the Eswatini problem, but it's a global a challenge. So this is in line with the universal health coverage objective number 3. So you are saying this NTB strategy serves as a blueprint for countries to reduce TB incidence by 80%, TB deaths by 90%, and also to eliminate some catastrophic costs. So it's very key to also understand that this document, it's not very big, but if you summarize it, you can look at the incidence, the deaths, and also the catastrophic cost. But we should look at our country context and also in terms of the laboratory, how does it relate to us? So in terms of the country context, as you can see in the previous slide, you are saying it's by 2030, basically it's by 2030. So we've got some data of how the country is actually performing. This was in 2000 and the data was from 2022, if my memory serves me well. So we are saying as a country, we had targeted that in 2020, we will have to reduce TB deaths by 35, but at least we have done ourselves to 58%, which is good and commendable. And we are saying we targeted 20% for incidency, and we did better in 2022 because we are at 50. So, which means as a country, we cannot fully say you've going to the right directions. There are always those gaps, but I think this is actually promising from a perspective of the targets. Unfortunately, there was no data on catastrophic cost, which we hope maybe the anticipate later on will collect. Okay, let's look at in terms of the lab colleagues, the lab and the NTB strategy. So in terms of the lab, the strategy is talking about 3 key objectives, which will be the main context of what you are doing in terms of sequencing. So we are saying objective number 1, you want to increase access to rapid and accurate detection of TB. That means there should be more cases which are bacteriologically confirmed, more adoption of molecular recommended diagnostics like GeneXpert, ultras, APAs. We are saying data connectivity must be always used. So these are the sub sections or areas that needs to be addressed. We are also saying we want to reach universal access to DST. So in this case, the WHO was looking at it in the context of rifampicin. So you can see there, I've made a question mark there to query I491F. So that's why please remember it because it's the basis of our presentation today. And also number 3 objective is to strengthen the quality of laboratory services. So that is also key in terms of saying all facilities, in terms of the lab, they must have a formal QMS. And we must strive to have them accredited in all the tests, be it GeneXpert, be it microscopy, culture. So these are the main lab targets and indicators. So basically, we have around 11 indicators, but we focus mainly on the objectives, which are 3. Just to summarize again, we want rapid and accurate diagnostics, we want universal access to DST, and remember the issue I491F, they want to strengthen lab services. So as a country, where are we? Can we really reach universal access to DST? Our interest, you remember, it's the main interest is rifampicin. Rifampicin being the main drug for first-line treatment and the most potent drug when it comes to TB management. So can we as a country reach universal access to DST? Yes, we can, but there are some challenges here and there. So I think you can recall that there was a question mark on my previous slide, which was saying I491F. So as a country, we have a problem. So the problem is there is a strain of TB that is circulating in our population. So this strain of TB, it will be like MTB detected by GeneXpert, but when you look at the results for rifampicin, it will say it's susceptible, but in actual sense, it's resistant. Why is it the case? It's because there is a mutation which falls outside the scope of or the detection limit for GeneXpert. So it's not just GeneXpert, but it's also LPA, and phenotypic DST. So we are going to go back a little bit in terms of the background. So we are saying all this information was found because the country took a step to conduct what we call a drug resistance survey for TB. So how are we really missing the strain of TB? So we can go to the next slide. Okay, here is a picture to summarize the reason why. So I hope you can see the pointer here. So we would say this is a rifampicin resistance determining region. So this is where in the RPO gene, so the gene expect will look from this area to this area. So basically this is like the whole codon or the gene for RPOB. So the gene expect will start from here to here. That's where it's going to focus. Then the mutation happens outside where the gene expect and also LPA detect. That's why then you will have cases whereby the rifampicin, because you are focusing on this region, we will think it's susceptible, yet in actual sense, it's covering some resistance because of this C491F, this I491F. So, like I have mentioned, so we got all this information from the drug resistance survey. So we are saying in 2009, when we did our drug resistance survey, and also in 2018, we did another 17-18, we did another drug resistance survey. So in 2009, we discovered that there were around 30% of the cases of MDR, which actually had this kind of mutation. That is from 2009. Then when we did another drug resistance survey in 2017-18, we found out in actual sense they were around the 58%, that's like 60%. They had actually multiplied, actually almost doubled. So this was very concerning for our health system because we're looking at a period whereby it's like less than 10 years, and we are seeing that the cases whereby we are missing, they've actually doubled. So that was a major cause for concern. So another point of note was that amongst these I491F strains, we found that actually 60% of them, 20% of them were actually having a mutation to the newly introduced drugs, which is bedaquiline and clofazimine. These drugs were actually introduced in 2000 and I think it's around 2016, if my memory serves me. But when it did our survey in 2018, we found that this kind of strain, okay, first of all, we are missing the rifampicin. Now we are saying it's more than just rifampicin. It's bedaquiline and clofazimine, which is a major challenge. Okay, so what did the Ministry of Health do? So in trying to address this problem, the Ministry of Health, together with its collaborators, including Baylor, so introduced, and also AHLS and TCP, they introduced a project which we call Sub-Saharan Sequencing Network that started from 2020, initial plan to end in 2022, and also expanded to 2025. So this project is supported by the Ministry of Health of Germany under the Global Health Protection Program, which is overseen by the Research Center Boston. But the main implementing partner here is Baylor. So in this project, we are working together with our colleagues from Namibia, also from Mozambique, here in Eswatini. So our colleagues from Kyrgyzstan, they are no longer in the project, but it should be very key to mention that our colleagues from Mozambique are also in the project. I think you will see later when we are talking about issues of surveillance. Here are the objectives. So we are saying under the project, you've got some main objectives. So our main objectives, I think, ladies and gentlemen, you can understand that most of the time, people complain about culture results taking longer, DST even taking the longest. So we are saying under the project, we want to strengthen culture-free diagnostics and establishment of a molecular surveillance approaches for high priority lung pathogen. So we are looking at MTBC and also SARS-CoV-2. So our main part of our main aims, we are saying we want to establish, evaluate, and also do some clinical translation of the NGS data for the detection of resistance to antibacterial drugs. So basically, we are saying here, we want to assist the Ministry of Health in strengthening the detection of detection of resistance or drug resistance, basically for MTB, and also to support the surveillance system. So we've got some key objectives. Whereby we are saying it's about strengthening network. We want to adopt some diagnostic workflows and also to focus on prospective pathogen surveillance. And lastly, we want the data that we generate from the project to be used or to be adopted in local guidelines. Okay, so what is sequencing? So I've given the background. So what is sequencing? So sequencing basically is to determine the linear order of nucleotide bases in a nucleic acid molecule, be it DNA or RNA. So here are the types. We've got Sanger sequencing. So Sanger sequencing, I think is mainly used for HIV. So you've got the next generation sequencing, which is the one that we'll be talking about today. Then we've got the third generation for, that's like Oxford nanopore. So by definition, next generation, as you can see, that is one of the types. So next generation sequencing, you're saying, is a DNA sequencing technology that sequences multiple small fragments in parallel to determine a sequence. So basically, ladies and gentlemen, if we can look at in the next slide, here, this is how the structure of the DNA looks like. So when we're talking about sequencing, we want to know that on the DNA molecule, is it A, G, T, C, C? Is it how the order is? So that order will tell us more information about the organism. So that's where we're going to derive our information in terms of the resistances, because we know that from the gene, it's going to be transcribed and then translated into proteins that will form a phenotype. So a phenotype can be resistance or no resistance. So we look at the order of the gene. So by sequencing, by next generation sequencing, we're saying the order, or in the small fragments of DNA, we will, all the fragments of DNA, we're going to look at them in the parallel manner. Unlike the other technologies whereby you just look one strand of DNA. Here we're saying all these species, we'll check the order for each and every one of them. So the general application for sequencing, so we've got the targeted sequencing, we've got whole genome sequencing, pharmacogenomics, genomic selection. So we're going to focus mainly on the first two there, which is targeted sequencing, the one that you are actually using for determining drug resistance. Then the whole genome sequencing will be used for surveillance. So these are the platforms that we use for sequencing. So we've got the iSeq 100, the MySeq, the MiniSeq. So all of these platforms, they can do sequencing. These ones are for Illumina. So as a country, we are using, I think we've got two of them here. So you can actually choose the last one, which is the biggest, but otherwise when you're choosing a technology, you have to choose based on your need, right? So for us, we're having the iSeq 100 and also the MySeq. So for sequencing, for NGS, the one that you are using in the lab, so using the iSeq. So what does the WHO say about sequencing? So ladies and gentlemen, the WHO has recently recommended sequencing technologies to be used for determining of drug resistance. So this is I think you can recall that you said the project started in 2020. So as a country, we managed to introduce this technology before the WHO even recommended it. So basically, these are the recommended kits. We've got the duplex MIC, which is highlighted here. This is the one that you're going to elaborate more on. Then the AMPO, Oxford nanopore, then the TPSEC. So our focus here today will be on the duplex, the one that you're using in the lab. So how do we select samples for sequencing? So we are not sequencing all of them, so they are sequenced based on priority. So this is like all samples which will be INH resistant or rifampicin resistant, and also treatment failures. So the treatment failures will only be sequenced only if the clinician has ruled out issues related to adherence. So maybe someone may ask, why are you only choosing INH or RIF? So we are saying here, let's start with the issues of INH. So we say we've got this strain, which is having the I491F mutation. So this strain was actually observed that it had the isoniazid resistance. So now we're using the isoniazid as a ruling test for sequencing. Then the rifampicin, yes, we already know that this patient is rifampicin resistant, but for patient management, we are saying we will need to sequence this strain to determine the rest of the drugs. You are going to see the capability of the TPS kit that you are using. So this is the basic workflow. So all the TP suspect from presumptive TP cases, we're doing our gene expect. So from gene expect, you will get your, let's start this side, MTP detected, rifampicin resistance detected. So we are saying we recruit because it's the project. You recruit, then you do the rest of the test and then NGS as well. So to simplify this side, we can say if it's rifampicin resistance, you just test with NGS, targeted next generation sequencing. Then when it's rifampicin susceptible by gene expect, we are going to do the other tests like culture, DST, line probability, or the XDR assay. Why do you do them? You want to determine mainly, is it isoniazid resistant or not, or rifampicin resistant? So if you're resistant, you recruit it to the project, and then we do targeted next generation sequencing. So please note that for our program, we're saying we want to start with targeted sequencing. Then all those samples that will qualify for targeted sequencing, we'll have to do a whole genome sequencing for surveillance purposes. So we always start with targeted sequencing. So this is the kit that we are using in the interest of time. So here is the kit, ladies and gentlemen. So the kit that you are using, these are the capabilities that it has. So from the kit that we use for sequencing, this side, these are the genes, related genes. Even here, these are the related genes. Let's start with the yellow section here for identification. So for species identification, we're using this target, which is heat shock protein 65, spolicotyping, that is the CRIPS. Then genotyping, we're using the phylogenetic SNPs. Let's go to first line. Thinking most of us, they do know the first line drugs. We're saying it's rifampicin, isoniazid, PZT, and ethambutol. So this side, RPOB is the gene of interest. For isoniazid, it's more than one gene that you're looking at, CAT-G, INHA. Then PNCA for PZT and EMB for ethambutol. So let's go to the red section this side. The red section this side. So these are the group A, B, XDR. All right, so this is a combination of both group A and group B. So basically, these are the second line drugs of interest. We've got fluoroquinolones, we've got bedaquiline and clofazimine, we've got linezolid. So RV0678, the gene of interest for bedaquiline and clofazimine. Why is it key for us, and also KGR A and B for fluoroquinolones, and also linezolid? So these drugs, they are except for protomanate because it's not here, but these are the main drugs that are forming the regimen for second line, the BPALM regimen, because you've got your bedaquiline there, you've got your protomanate, unfortunately it's not here, it's not yet part of the scope. You've got your linezolid, then you've got your fluoroquinolones here. Then the group C, the kit still detects some of these drugs, amikacin, streptomycin. So these are the injectables. So it's part of the kit, even though we're no longer using them, but you'll get the information. Geprimacin and kanamycins, you are still getting information about them. So in just one run, we're able to tell whether the patient is susceptible to first line or second line drugs. And most importantly, we can tell that which lineage is this particular strain that the patient is having. So this is the basic workflow that we have for sequencing. We always start with DNA extraction. The DNA extraction can be from sputum. We do process sputum or from the cultures. Then we do the target enrichment, which is the duplex PCR. Then we do libraries, because in order to sequence the samples, we need to have libraries. Then we have this stage, which is the actual sequencing. The actual sequencing is the one that will take place in the machine. I think I'll show you the pictures later on. Then you've got your data analysis using the duplex web. So this is the cartridge that we use for sequencing. So the sequencing, it takes place here. So the cartridge basically has the reagents for sequencing. The cartridge has the reagents. Then the sequencing takes place on this small glass here. This small glass gets here in B. Then the sequencing will take place. So once we finish sequencing, we have our data. So the data is like called FASTQ files. So this is still the basic workflow. And then here is expanded to say under data analysis. Once we get our files, which we call FASTQ files, we upload them on the duplex web, and then it does some analysis. I will show you the results just very soon. So this is how the duplex web look like. So you log in and then you put your password, then you upload the sample for analysis. So this is an example of the results that you get from our sequencing. So here you can see that we have all the drugs like we mentioned, from rifampicin to clofazimine and bedaquiline. So there are some sections here, like for instance, RPOB for rifampicin. It does state INH here, this side is the type of gene that is related. So basically, let's just look at saying, it will tell you this is the organism that we have. Use this polygotype, and this is for lineage analysis. So what does this mean? If it's red, it usually means it's resistant. If it's blue, it means it's uncharacterized. If it's green, it means when it's green, it's saying I did not detect any mutations which can confer resistance, which you can translate to susceptibility. And also it does provide some quality scores here. It goes up to 3+. Okay, so once we get, looks like some of my slides have a problem. Okay, as a gentleman, then once you get our results from sequencing, we send them to the clinical advisory committee. The clinical advisory committee assists with further interpretation, and then they share the results with the treating clinician. So basically what they will share is the recommended regimen. Because once we get the results for sequencing, we will find that there's a need to change the regimen. So the clinical advisory committee will then recommend the regimen which will be used for that particular patient. So I would actually go through the whole process here, but I just prefer to summarize it. So you have the data from the NTRL, we process the samples, you have the samples we process. Once you process, we gauge the results are then given to the CAC. The CAC, they will review and then come up with a specific regimen that will be used for that particular patient. So let's look at this particular graph. So this particular graph, we are trying to show the importance of sequencing. So the first part here, the first column is expert ultra. Then the second one, the FL means first line, LPA or line proper say, FLDST is phenotypic DST. So this R is for rifampicin, then the H is for isoniazid, right? So the green, remember we said if it's green, it means it's susceptible. So for this particular patients, we did sequencing. Then we compared the results between expert ultra, line proper say, first line, and also first line DST. So we can see that from the first column is green. That means by expert ultra, the patients were deemed to be susceptible. We did our line proper say, first line LPA. When we did our first line LPA, this one gives us the results for rifampicin and isoniazid. So we got these patients to be rifampicin susceptible. But these patients were also having some resistance to isoniazid. You remember we said isoniazid is one of the key inclusion criteria. You're going to first-line DST, it also gives you RIF, INH, and your ethambutol. Even by DST, our gold standard, most of the samples you can see that they are green, but they have isoniazid resistance. So now, our first three tests that we are actually using on a routine basis, they're saying these patients are actually, they're saying it's isoniazid monoresistance, basically. When we did our sequencing, which is the biggest section, our sequencing here, the first 5 here, they are all red. That means for all these patients would have been actually treated as drug susceptible TB. But once we did the sequencing, we observed that I know these patients actually have resistance to not just to isoniazid. You've got your rifamycin is resistant, PZA, you've got the ethambutol, streptomycin, and most importantly, they also have resistance to, most of them have resistance to beta-quinine gliflozin. So that leaves us with a lesser choice when it comes to regimen selection. So why is it so? So all these are the strains which are having that mutation, which is the RPOB I491F. Okay, so far, since we implemented our sequencing, just to share with our colleagues, there is this paper on stool sequencing using the duplex. So basically the sequence or the duplex kit is not only used for filter, you can also use stool for sequencing. Then we come to this another, this was an abstract which is currently being worked on to become a manuscript. So now how do we plan to optimize our workflow? So we are saying here, in our facilities, we have introduced the expert for stool. So we're saying facilities should bring the stool samples to the NTRA so that you can be able to detect this I491F. You remember stool samples cannot be cultured, but stool samples, you can have sequencing on stool samples. So we want to detect this I491F from the pediatric population. And another important thing you are saying to facilities, always collect the two samples. GeneXpect is fine, you will do your ultra, it will say susceptible. Use the very same sample for XDR to be INH resistant. So for you as a clinician, you must be very curious now to say, is it not possibly this strain that the sequencing guys are talking about? Hence, it's important that you always bring the other sample for culture sequencing and drug susceptibility testing. So we are currently working on the report, and most importantly, we are saying sequencing must be fully adopted for clinical management, into our guidelines for clinical management. So we need to do some more trainings. We do acknowledge that we still need to do some capacity building for our clinicians. You remember that we said currently we're having our clinical advisory committee, but through full implementation, we would have to have clinicians being able to interpret the results for themselves. So we are also focusing, or we'll be focusing mainly on surveillance going forward up to 2025. So not only for MTB and other lung pathogens like your SARS-CoV. So here is our summary. We're actually at the end of our presentation. So we are saying Eswatini is one of the leading countries in adopting TB diagnostic technologies or methods. So we are saying we introduced sequencing in 2020, 2021, but you can note that it was recommended 2 years later by the WHO. So we are also saying genome sequencing is a great technique for genome-based surveillance and rapid resistance prediction. This is evident through the issue of I491F, because you won't get the resistance by expect, but you can always get it by sequencing. So this approach or this sequencing technology will help us to actually reach our main target, which we are saying, the universal access to DST. Not just for the few, but for everyone. So we are saying POB I495 strains are potentially spreading across borders and evolve towards XDR. So this is evident through the inadequate traversing resistance that I talked about. Then you are saying further efforts in programmatic implementation of NGS and WGS-based surveillance is needed. And then lastly, you are saying, ladies and gentlemen, if we do not sequence most of all our strains of MDR, we are going to miss, we said 60%. We are going to miss 60% of the MDR cases. So all these 60% you're talking about, if we don't get the assemblies to be sequenced, they will be spreading the organism to the next susceptible host. Okay, this is just for information purposes, in case you have got some questions. Yes. Then, ladies and gentlemen, this marks the end of my presentation. We'd like to appreciate everyone here on the slide, the MOH Baylor Foundation Research Center Postel, while managing the project, EHLS, NTCP, the NTR, EMDC, and also the Public Health Lab. Also, our implementing partner, ICAP, and all other implementing partners, because everyone plays a role when it comes to ending the TB epidemic. And also, I would like to thank you for attending today. Thank you very much, colleagues.
Thank you so much, Mr. Ziani, for that comprehensive presentation. Yeah, it was really an eye-opener in terms of where we are in the NGS project and also aligning that into our TB diagnostics. Thank you so much. At this juncture, colleagues, we'd like to take a couple of comments or questions you have on the already done presentation. And Mr. Ziani will take all the questions and respond as per the need. You can raise your hand, colleague, if you have a question or comment, then we can gladly take it. We also have a chat box. Maybe if you're not comfortable raising a hand, you can write your question right there and then we'll gladly respond to it. Okay, I take it as everything was well, loud and clear, and well said. Thank you so much, Magaliso, for the presentation. And there was a comment from the box from Mr. George requesting that maybe I go through how we can access the previous presentations and the links. So can I share my screen once again so I can just navigate and present to you how you can access that? Okay. It's pretty simple, colleagues. Can you see my screen? You can go to YouTube and then just type Eswatini medical and dental cancer. And dental cancer. And then search. And then here you'll find this page. Just click on the first one, which is Eswatini medical dental cancer. So here, this is the page we thought we'd create to make all the videos accessible to everybody to access anytime and anyhow. So here you'll find all the videos that have been uploaded. But what we did to segregate between the cadres, we thought we would make maybe a couple of playlists. We have the medical and dental cadre. This one is specifically for the doctors and the other cadres, CME recordings. And then here, these are for the lab. So if you feel like you want to access any content that has been played before and just to learn and also gain more knowledge from that. So you can access like this one was presented 15 days ago, which was our 6th CME. So under the description, you can find the date which was shared, and then also you can find the CPD activity number, which in this case is P001 2023 TS2020. And then this is the link to the quiz, like I said earlier, that you can access once you watch the full video and learn. I advise that please go through the quiz. It's just 5-10 questions generally on it, and then it also adds our attendance register at the same time to also keep track of people who are also learning more to that. So we created this platform, colleagues. We do hope that as scientists we can continue learning, and then also we avail physical trainings, working with all the partners and also the government to avail trainings which are aligned with the CPD program as well. So the idea is to help everyone at whatever level, could it be a pathologist, technologist, technician, even lab, to continue learning and also advancing our skills into that. And lastly, we also encourage, online training that we get from outside the country, or maybe out in online learning platforms, which are CPD related and also advanced knowledge as well. So colleagues, just like any other session, we usually, at the end of the presentation, we usually present just a small quiz for you to go through, and then also which will also test your level of knowledge retention as the presenter was presenting. So I will present here the QR code. You can scan directly from the screen and try to access the quiz. And also on the chat box, I will also paste the link as well for this quiz. Please go through it, and then you'll mark as your attendance register for the session today. Yeah, you can find it on the chat box, colleagues. And then maybe while you edit, I would like to appreciate colleagues for joining on today's session. You can go through the link and then complete, and then we'll tell you your score at the end. Then after a while, we'll send to you directly through your email, certificate of attendance for this session for today. From my office, we thank you so much, colleagues. Please take your time and complete the quiz, and then we'll meet again on the next session, which will be in a week's time. Thank you.